Well, now that we've worked through the whole process of reading the DNA, making messages, converting over that to polypeptides, and ultimately to proteins that give us phenotype, we want to back up a little bit and talk about mutations. So we're going to go back to the DNA level, but we're going to talk about it in terms of what it produces. Obviously, the types of effects it can have on phenotype, which means we're going to talk about the effects it'll have on proteins. So this first part, I just want to do an overview, a bit of an overview, and a couple of the primary types of mutations we might see. And then the next few lectures, we're going to talk about more specific forms and other things. Now, we've already talked about the fact that mutation, I mean, we're going to take us way back to this whole process of domestication. Remember, we're cycling back now toward the earlier parts of the semester. And we remember that once we domesticated organisms, we started to see variants, right? And it would turned out that these variants represented what were mutations, very often mutations that were in the natural population, but would become obvious once we started to inbreed and use a small numbers of animals. Some of these were good mutations, some were things that we didn't want. But if we wanted them, then we would tend to break them apart and use those as our sort of our ideal. And of course, Mendel, working on peas, was working with mutations, his smooth versus his wrinkled peas. You were tortured for many lab exercises working on flies of different types. Well, these are mutations, many of which were produced by radioactivity, purposely produced. Others were naturally occurring in forms. Now, whatever you're looking at here, though, remember that we said any genetic material, just go back and kind of refresh us, whether we're on this planet, this universe, wherever we are, and whatever the source of the genetic material was, we have some basic must-haves, right? And then these were the must-haves that Watson and Crick had to argue for when they were trying to design, uh, illustrate the model for the structure of DNA and how that could serve as a genetic material. And we know well what these are. These are obviously they must be able to transfer effectively generation to generation. They must store vast amounts of information, and we know that our DNA is capable of doing that. And they must have replication that has high fidelity. Now, notice this high fidelity part here. Uh, Replication has very high fidelity. Billions of bases get copied perfectly. However, it's not 100% perfect. And that lack of being 100% perfect often leads to having mutations. Now, it's good that information can be mutable at some levels, right? Because we want it to change. The environment is never constant. The sources of things that come in exposed to an organism are never constant. So there needs to be variation there in order that the system can maintain itself in these changing environments. And of course, we can always make fun stuff out of it. You know, mice that glow in the dark, different colored fishes. We can play with it, and we certainly are doing that. And these are all mutations that are being produced in various ways. Now, in the real world, you know, mutation rates and understanding that are very important. For example, HIV, just one example of a major disease that attacked a large part of the human population, caused great amounts of issues, including death in many situations. Just going back and trying to figure out where these variants came from. Turns out to be quite a few variants. Some came out of chimpanzees, and that was a completely different form, it turned out, than those that came out of the green monkeys and other forms over here. So what? Well, if you're trying to treat these and develop pharmaceuticals for these, you kind of need to know what the mutation, where the original forms were that will help you develop these forms. And another really important thing about we see about HIV, and one of the reasons it's an example of some of these very, very dangerous pathogens, is that once it enters the system, it mutates itself. It continues to mutate rapidly. So it's not just mutating out here in these host organisms. Once it's inside, say, a human, you see that there's mutation that can occur in the lungs versus the spleen versus all these others. So, and some of these mutations make these particular ones, you know, resistant to some of our drugs and things. So it has a very high mutation rate. And even once it enters the organism, it goes to the specific sites, it mutates, which makes it very, very difficult to try to completely cure or eliminate, even though there's some very modern approaches that are certainly helping increasing the longevity of individuals with HIV. Another example was Ebola. It was a big scare a few years ago. Okay, and Ebola still exists out in the natural environment. A terrible disease. Basically, just once it hits, it just takes the body over and causes death for sure in a very short time. Very few people survived these Ebola outbreaks. Um, there have been over 400 known mutations of just the Ebola strains that we know about, and there are probably more Ebola strains out in the natural system. Okay, and one of the reasons for that is viruses do this replication, but remember they're replicating generally inside of a cell system that's not their normal cell system. They're taking over other cells, and they often make very bad copies. So when they do that, a lot of them become bad copies, so that the virus isn't functional. Other times it becomes a copy that even makes it more dangerous and more contagious. Like what would happen if Ebola became an airborne virus, right? Instead of one that's transmitted primarily by touch or fluids type exposure, right? Just to give you some examples of this is the Ebola rate right here, or the mutation rate that we know about. And it's up there with a hugely phenomenal rate. I mean, even influenza and things, flus down here, it's much faster evolving than those rates. It was mutated, just hugely fast. And influenza is the flu, right? Common flu that we have, which we have to have a new uh, vaccine every year just to kind of put up with it, right? To deal with it. Well, this is even faster than that. And so you see these various sort of peaks in the form, and these were peaks in mutation rate of this disease. So once it was in the population, it just very quickly started to mutate. And one of the things that's really important about it, and points out here is, and we will get to this when we talk about population genetics later, but it's really dependent on the size of the populational pool where it exists, right? So the more people that become infected, the more mutations are available out there. And so, you're trying to curb that distribution is really difficult. So the more, basically the more people that became infected, the higher rate that mutation showed up, because there was more chance, more replication, and more chance for it to go on. Well, it doesn't always have to be bad, obviously. With the reason we have so many different forms of proteins and other things is very often because we've had these duplication events, which allowed mutation to occur and change them and specialize them. This is a myosin family tree here, if you will, of the proteins that are all involved, all the variants that are known. For us, we see some in smooth muscle versus the difference we find in cardiac muscle versus those that are specialized for skeletal muscle. There's also those that are specialized for plants. There's all sorts of forms. Now, these all come from a central part, and they represent mutated versions of this gene that have gone off and become, useful in their whatever cellular environment that they've been put into. So it's not all bad, right? This is one of those, we talked about these multi-gene families, right? Or when we talked about gene structure and genomic structure. Well, this is a way you can build a multi-gene family, is by having these mutations. Those mutations become functional in a particular cell type or particular tissue type, and they are more functional there. They maybe require less energy to do their catalytic activity, or they just fit into the system better, or whatever. And so they become positive things when you have mutation, right? Now, let's talk more about mutation in general. Okay, so just remember, we just talked about proteins and protein structure, that the amino acid sequence there and the interconnections that are made between those are really what drive shape, right? So the structure basically builds shape, and shape is what makes a protein function. Well, guess what? If you mutate one of these systems, you change one of these amino acids, if it's the right time to change, it will end up in a mutation that may potentially affect the protein itself. Some are silent and they don't really matter. Other mutations are, third position and they don't get seen. But if you do change an amino acid, and particularly if it's in a place where like these cysteine disulfide bridges and places like that, you can completely change the structure of the protein, and therefore you're going to change its function. Sometimes for the better, that's fairly less common. More often for the worse, become malformed proteins, but form. Now your book lists a serial number of these, and it's a nice listing of all the types of mutations that are covered in the book. And many of them, we've talked about lethals before, right? We're going to talk about some others of these. Now, we're not going to do all of these. They're well covered in the book, but we will talk about sort of a large section of them, particularly these that are transitions and transversions and frame shifts and things that you should know about. They're very commonly named, okay, and silent mutations as well, missets. Okay. Now, some of these overlap, like this one that's loss of function. Well, a loss of function is because you had a missense, right? So those somewhat overlap, and you have to think about it in that terms. But these are common terms that we see, and I think you should go through each of these and learn them on your own if they're not the ones that we cover, because if they're not used here, which they will be on exams and things, but even if they're not used completely here, they'll be used in other courses and are really important to know. All right, so let's think about the ones we are going to look at. We're going to look at the primary types of mutations that are either transitional or transversion. That's what we're going to do in the rest of this lecture. In the next lecture, we're going to pick up on other types, what it means to be silent, what it means to be a missense, what it means to be a nonsense mutation. And then we'll talk about frame shifts, both in frame and out of frame shifts. Okay? But for now, we're going to concentrate on these first two and talk a little bit about those. Those are kind of general categories of mutations that can lead to these other outcomes. Okay, so they can have a transition that's silent or a transition that's nonsense or whatever. Okay, so just simple definition. The transitional mutation is when you stay in the same class of the base that you started with, right? In other words, a pyrimidine stays as a pyrimidine, a purine stays as a purine. So there are 4 possible base changes that allow that. You can have your pyrimidines of T changing to C, or a pyrimidine of C changing to T, or you can have your purine of G to A or A to G convert. So 4 possible outcomes there. Okay, they're often shown in little charts that maybe a numeric chart might look something like this. People will draw those up. Sometimes they're little boxes with A, T, G, C, and then you go across or down. But they're basically these little possibilities that you can have here, and they're 2 arrowed, so that means that they can go either direction back and forth. So there's 4 possibilities here. Okay. Now, transversion is just the opposite. Transversion is when you go out of class. So you change a purine to a pyrimidine or a pyrimidine to a purine. And as you might expect, there are 8 ways that you can do this. Okay. You could go A to T, A to C, or C to A, or C to G. You've got all these possibilities. So there's 8, A to C, A to T, G to C, all these forms. Okay. So you have each one of these variations that you could have a simple change. Thus, those would be a transversional mutation. Okay? All right. So we have transitions and transversions. Well, it turns out that changing out of class, going from a pyrimidine to a purine, requires more energy and in fact is more readily detectable by the editor system. So it turns out that transitional mutations are more common than transversional mutations. In fact, when we set up and we try to do those little trees like you did in lab where where we just examined the number of changes between a couple of sequences, we said, okay, this is how many, just count them up, and you can build a little phylogeny or a little tree showing the relationships based on that gene segment. Well, there was a question in that one of those labs that said, well, what else could you do? Well, one of the things you could do is, in fact, look at whether you had a transition or a transversional change. And then you can weight those based on their probability. For example, some of the regions in the mitochondria, that maybe the cytochrome B region, the transition transversion ratio is around 13 to 1. But you'll see if you compare across a number of samples from a number of different species, about 13 transitions for every 1 transversion you have. Some places are 2 or 3 or 4, others are up in closer to 20. But whatever those numbers are, you can use them as a modifier to put weight on transversions. If you see a transversion, it's probably more of a more powerful statement about there being a difference than if you simply see a transition, because you're going to see a lot more transitions. And these occur in very different, patterns. Mitochondrial DNA, remember we have circular mitochondrial DNA, about 16,000 bases. At KB is per kilobases. Very commonly in that, you see a lot of transitions between C and T. And actually, you see quite a few between A and G. Much less common is things between our transversions between A and C. These simply don't occur in the process as much. They do occur, they're there, but they're not. Notice this is about a 32 to 1 or 2 ratio. So that's a 15 or 16 to 1 ratio for that particular change versus this other one. Now we go into the nuclear region, we still see that transitions occur more commonly, right? Certainly more than some of these others, but they're not at this at a much higher rate. Okay, it's usually in nuclear DNA, it's maybe 7 times, 9 times, maybe up into 10 or 12, 14. But it's and it can be quite large, but they're not common. The point is though, you do see more transitions than you see transversions. Okay? And then not only do you have these differences, but you have spikes in time when these occur. So if we start looking over long periods of time, it turns out that we can see that there's mutations, that certain types of mutations become common in organisms, and then they sort of become less common. This is just an example of humans, and this is converting from a TCC to a TTC mutational fraction that someone looked up. And these red areas represent areas of hot spots where you had lots of this mutational change coming along. One was obviously up in Europe, up in here, and the other spot was down here in Southeast Asia, Southern Asia, right here. In Africa, during this period, you didn't have as many, and over here in Eastern Asia and other areas of China and places you didn't, from those populations, there's no evidence. But there was a long period of spike right there. And actually, the biggest spike occurred about in human populations about 12,000 years ago. You saw a mass accumulation of these mutations. And then again, there was another spike about 2,000 years ago. And then so you had these pulses of when you would have these mutations as in this case, when humans moved and made major changes, or when technology changed and came along. So it's something we can look at and look at the difference between these types of mutations and how the rates change. And then they can be useful for even going further back, millions and millions of years. We can look at the differences between extant species, so things, gorillas and chimps and humans and orangutans that are around now, and backtrack and figure out what types of mutations have been the most common in those lineages, right? And this one, the orangutans looked like there were more transitions at these particular C to T type transitions. Once we started branching off over here into the great apes, we had more AT originally, transversions. Were common than they were in other parts of the tree, is what it means. And then eventually there was these other types of transversions that separate out, say, the chimps from some of their close relatives, or the gorillas from humans. Okay, so we can actually, they're used in these trees. In fact, when we build these topologies, one of the things we try to do is figure out exactly what base changes, differences have occurred between the DNA, and then we come back and we look at these models about how the rates of those and functions, and then we put in these mathematical models to help us stabilize the tree, make it more informative, etc. Okay? Well, those are the two biggies for us. They're transitions and transversions. And now we're going to come back and say, okay, if you've got a transitional mutation, well, so what? What does it do? Okay, and that's going to be in the next lecture when we start talking about silent versus missense and nonsense, and some of our other variants that we would have of that.